more patterns. So first of all, welcome everybody. Third lecture, second real lecture. Last week we, got introduced to the topic of patterns and pattern mining, and specifically pattern set mining, right? The idea of using compression as an objective function to find a set of patterns that together describe the data well. We saw that these give insight. We saw that these can be useful, right? We saw it for classification, we saw it for outlier detection. We didn't see it for clustering, and unless I miraculously have a lot of time left today, we'll postpone that or leave it for what it is, but believe me, you can also do clustering. Today, I want to dive a little bit deeper into patterns, but also bring us a little bit closer to the now. Because most of the topics that we saw last week were, a bit more dusty, if you wish. I mean, fundamental and very necessary to know, right? As necessary as, let's say, linear regression. It's also dusty as hell, but if you don't know it, yeah, you're not going to have a great time in machine learning land. So today I brought 3 topics for you, and I'm going to present them in order, and each, bring us a bit closer to the now, and all of them are connected to what we've been talking about last week, and also connected in between. I'm going to point out some of these connections, but not all. So this is again a task for you, right? Okay, what the hell is he talking about, and what has he been thinking? Well, sort of like an Easter egg hunt, right? Very fitting to the time of the year. So now my goal today is that at the end of the lecture, you now not just know from last week what are patterns in their basic form, but what can you really use them for? And for tasks that otherwise, I mean, how would you even solve them, right? That is one of the aspects. And at the same time, what are then modern applications? And modern applications that you would not be able to solve otherwise. It's just, you need this idea of pattern mining, otherwise you're not going to have a good time, right? That is my goal with these 2 lectures. Good. First, we got in our time machine, 5.5 years ago, 2019. We're in, where were we again? Würzburg. We're in Würzburg. Jonas was then still a PhD student. I mean, then afterwards he became a postdoc at Harvard. Meanwhile, he's a research group leader at MPI, so he's not stupid. I think that is fair to say. I mean, he's also a very nice guy on top of that, which. And this was his first real paper of his PhD. We started at the very beginning of his PhD with the idea of like, okay, hey, there's crimp, there's slim, and they can find patterns. These conjunctive patterns, A and B and C together happen somehow significantly often, significantly enough for us to put them into the model, something like this. What about rules? What about association rules? Let me show you. Let's first explain once again, because I'm using the slides that he used back then to address a crowd of 400 or 500 people. It was his first presentation. I mean, he might have been a little bit nervous. I'm not sure if he's willing to admit, but he might have been. And I'm using exactly the same slides with a slightly different story, right? But this is both, not so much because I'm lazy, but also to give you a bit of insight, not just what does an actual research paper look like, but also how are these things presented, at what level of detail. This is useful not just, because these slides should be didactically good, otherwise we made a mistake, but also if you ever give a presentation yourself, a style and a depth of these 3 presentations I give today could be something that you aim for, right? Everybody has their own style, but keep this in mind, right? I mean, I have, let's say, layered didactic goals in my lectures. So here, let's explain again what is all of this useful for. Jonas is a bioinformatician, so he likes thinking of finding patterns in genetic data. Here we have SNPs. SNPs are single nucleotide polymorphisms or point mutations, right? If we have a reference genome for a particular organism, here patients, so let's assume it's humans, right? And we sequence, let's say, your DNA, we can see, okay, in which points do you have a genetic variant? SNP, right? But at one point, a difference, that's a SNP. And what we can then do for each of you, compared to this reference genome, figure out, is there a difference on which spot, yes or no, right? And hey, we have a binary table. Well, we know what we can do with a binary table. If we have such a binary table, it could look like this. This is, of course, a comic version, right? what do we see here? What can we imagine that might be going on, and what might we want to be able to extract from this data? We see that SNP A, so on location 5, this happens quite a lot. We see that here this green block, they seem to, well, group together, right? But there are some errors in between, right? Then this red one over here, yeah, is on the one hand you could say is really independent, except that, hey, look, the blue and the red one, they are almost, perhaps even exactly, can't really see, actually from this angle can see it, they're exactly mutually exclusive. That's another type of pattern, but not a pattern that we could grasp so far, right? It's more like if this one, then not that one, and vice versa. We don't have the language yet, right? We also have these blue ones over here, where these two C's, they also seem to happen jointly. Now, if we would apply traditional pattern mining, or even the tools that we saw last week, what would we get? We would probably get the bluish pattern, right? Because they happen closely together. The red one, yeah, we would see it as a singleton. It would be like, this is noise. I can't really say much there, right? This blue one, well, it could well be that we get it together with the green ones. Because, yeah, so let's call these just A, B, C, D, right? The first 4 columns. They happen quite a lot together. However, these noise things here, they might even screw up. They might, yeah, so right now there's relatively few of these holes, but if there's sufficiently many of these holes, Slim and Crimp can't really deal with noise so well. So they might not even be able to find that block. Do we want to find that block? Or could we perhaps describe the structure between these first 4 columns differently in a more compact and more interpretable, insightful manner? What would be the wording you would choose? Would you say that, well, A at 5, T at 100, G at 103, and G at 155, that is the best description for the structure we see around A, or these first 4 columns? Or do you see something different, maybe? Yes. So if you allow me to rephrase this, that if we see an A, there is a much increased probability for seeing, well, TGG, right? So an increased probability. People who buy diapers don't necessarily always buy beer, but there is an increased probability. I now recall that last week I didn't even explain why on earth there's so many beer and diapers on my slides, right? Is it not because I have some weird fetish? I mean, I hand on heart, right? I mean, I promise. Everybody's weird, me included, but not in that direction. No, actually, the frequent pattern mining stuff, way before the stuff that we discussed last week, right? The pattern set mining, really the traditional stuff. Now the name escapes me. Really big guy from Canada. Anyway, he used association rule mining, frequent pattern mining, on data of a large supermarket chain in Canada. And one of the patterns they found, and that were surprising, but also like, well, with very high frequency, was beer and diapers. Which, like, right? I mean, that's, again, so lots of customers had a strange fetish, right? So they looked into it, and their explanation for this particular pattern was that, okay, young family, right? I mean, that's, they run out of diapers, then the young dad gets kicked out of the house to buy diapers while he's doing shopping. Like, wait, we're also out of beer. So not everybody who buys diapers buys beer, but there is a strong correlation. It's not the other way around, that you sort of buy beer like, okay, I'm going to get really completely shit-faced. Let's also get diapers just in case. That is not the explanation they came up with, right? But yeah, true story. Or at least the story is true. I'm not sure if the facts are true, right? I didn't look into them myself. Anyway, but that guy, Brendan Kitts, he made really a ton of money with doing this type of market basket analysis for a number of supermarket chains. I mean, this is what he did in the 90s, right? I mean, so now we're a little bit late, perhaps, but anyway, that's the beer and diaper story. Good. So this is then with patients and SNPs, but, well, in modern bioinformatics, we're actually looking at single cell sequencing. So what is the genetic makeup of an individual cell? Because every cell is a bit unique. And then what we know is that through epigenetics, basically small modifications, let's say live modifications of the chromatin, which are called methylation, that either close the chromatin or open it. And if it's closed, it can't be transcribed so well. So it can't be expressed. So you may have, let's say, a bad gene, right? I mean, that might lead to a bad disease. But if the chromatin is closed because it's methylated, who cares? However, if the opposite happens, right? If the good genes get methylated and the bad one doesn't, then all of a sudden you have a lot of expression of the bad gene. So just looking at what is happening at the chromosome level is not enough. You need to kind of look like, how is it being used? Well, that is what single cell sequencing allows you. Then we can check whether individual locations are methylated, yes or no. And hey, look, the data looks exactly the same. And we can also do supermarket basket data. I mean, that's good. In general, we can speak about samples, features. I often speak about items rather than features because, well, these are items in the supermarket, right? So this is just how my brain has been conditioned. And these can be samples, can be records, can be transactions. Doesn't really matter. So I already discussed a little bit what are now these different types of structures we could find, right? So if we now plot different statements that we can come up with, and this is not complete, right? We can do many, many more things, but we start from very simple and then all the way to something that is super complex, right? I mean, we call it voodoo, right? And basically a neural network can express anything, right? But we have no idea what the neural network is expressing, right? I mean, so this is a bit of a balance that we need to find. At the very low complexity end, we can have just basic atomic operations, such as A and B, right? Beer and diapers. It could be that, well, people who buy diapers tend to buy beer. Could also be, right? I mean, or this gene tends to activate that gene under certain circumstances, right? You could sort of read the A as the circumstances under which we then see the B and possible other things. You can also do mutual exclusivity, like we just saw, right? I mean, with the red and the blue one. You can do combinations of the two. You can imagine a version of Slim, Crimp, whatever, that can do both the conjunctive patterns and rules, perhaps even rules over conjunctive patterns. You can do complete hierarchies where you have like, well, A and B XOR C. So it's basically this gene is on or that gene is on, because if you have neither, you're dead, and if you have both, you're also dead. So you need to have one of those, right? I mean, together with mutation A, and then, I don't know, I mean, you have reddish hair or whatever it is that this encodes. You can see that you can express more and more things with such a, well, on such a dimension, right? I mean, and this is far from complete. Now, the interpretability is another angle that we need to consider. These very simple statements, they may not be able to express all the structure in the data that we would like to be able to recover, but they are very easy to interpret. These hierarchies are already a little bit less interpretable because, okay, what is now an AND with an XOR or an XOR over an AND over an XOR? This can explode relatively quickly. So you may want to restrict the depth of this hierarchy, right? Good. And neural networks we're going to be talking about later. People have, of course, been looking into these things. Right? So, for example, association rules, we were not the first ones to think about this. In fact, association rules, the whole idea of like, okay, diapers tend to, people who buy diapers tend to buy them beer. That was the original motivation for mining frequent patterns to begin with. Because mining association rules, if you think about it, is actually quite a tricky problem, because which sets should be the conditions of what other sets, and it explodes really into your face. However, if you say, hey, I want to have dependencies that are strong enough, then we need to see both the head of the rule and the tail of the rule often enough together. If I see the head very often, but the tail never, I'm not interested in the rule. And obviously that means that head and tail are also going to be, well, at most as frequent as then the tail itself, right? So this is why people were looking into this already in the early 90s, back when you could make money with this. The conjunctions, I mean, then people dived in much further. Those, as we saw already last week, actually are not so interpretable after all. They're not so expressive, but you tend to find billions of these things. And yeah, well, you need a very big stick to shake at before you can find anything. Then people started thinking about statistical testing. and I skipped over this last week, right? But rather than doing this compression game, you can also think, okay, for each individual pattern, is the frequency, how often we see it in the data, is this significant with regard to some null model? For example, is the frequency of seeing A and B together, is this significant compared to how often I see A and B just individually? And then you can devise some test, a chi-square or an independence test, lift, whatever, right? That sounds great. However, in practice, you're going to find billions of significant patterns. Exactly the same problem as what we saw last week, because if, let's say, A, B, C, D, E, F, G is the true pattern, then all of its subpatterns are also going to be very frequent, and all of them are going to be significant. So if you test things individually, you're not going to have a good time. Which is where then the compression-based approach, they win in a way, because they don't look at individual patterns, but rather look at the complete set. Well, these methods, so the first one we've seen, this is CRIMP. The second one is a paper I wrote with a good friend of mine, Nikolai, and there, we have a dashed line because what we actually find are decision trees, a set of decision trees that encode the data well. We had special reasons to do so. And if you have a decision tree, if you take now the path from the root over all the nodes to a leaf, we actually get a rule. If A is 1 and B is 1, then, C tends to be 1 or 0, whatever, right? So we can interpret all the paths in the tree as rules. It's not entirely fair to say that PAC, as this method is called, is really a rule miner, because it's more like we're torturing it to confess rules where, but you can interpret it. We're going to be using that later on in the evaluation. So let's look at the problem that we wanted to solve. We have a good solution for these conjunctive patterns. SLIM is pretty decent. And we've seen diff norm and some advances, but let's stick here. What we want to do is actually expand this to rules, and not just A to B, but A, B, C to D, E, F, right? Arbitrary length and whatnot, right? And actually we want to have combinations of the two. This is where we started pretty much at the start of Jonas's PhD. How hard can it be, right? We can already do the conjunctions, right? A increases the probability of B. Come on, right? How hard is it? And especially the combinations then. It's harder than you think, much harder. We banged our heads against the wall for quite a while, but we cracked the nut, not our skull. But we had to really think about what does it mean that it's a pattern and not a rule, and vice versa. How do these two now connect? Because we will see in a second. For example, this data, this toy data, here, intuitively, we say, and perhaps you agree, that the correct model for this data to describe it is that A towards B, right, and A towards C. If I see an A, this increases the probability of B, and independently of that, A also increases the probability of C. We could have, of course, said, okay, well, actually, this is A and B, and A and B and C, and A and C. This is another way to describe this data. And depending on how you craft your description language, it may be equally compact, but for this data, I would rather have this than these three individual patterns, right? I hope you agree. Now for this data, and I should make a mental note that next time I animate in the rules afterwards, because they can ask you. Here I would say that A increases the probability of B, and B increases the probability of A. Possibly, A and B increase the probability of C, but we don't really have enough evidence to decide between those two options, right? But you could also interpret this as A towards B and A towards C, right? The same model as we have here on the left-hand side, but somehow it doesn't really feel the same, right? Here, these rules we can interpret as independent of each other, more or less. And here, not. And that's what we want to express, right? Here, this is quite a clear case. This should be A towards B and C. This is where we had trouble for a while, because intuitively I would like to say this is the pattern ABC. But the pattern ABC is also the perfect rule A to BC, BC to A, and so forth. So, and in the end, we settled on the philosophical notion, no, actually this is empty set to ABC, And with that notion, we could then craft a score that also favors this as the result, rather than any of the others, which if I were to get in this setup, this answer, it's not wrong, but it's a bit, well, confusing, right? I mean, that's, I might say, okay, I'm going to put an advertisement on A, right? But yeah, maybe something happens on B and C. I don't have enough evidence, right? I don't have enough evidence. And above all, which we now swept under the carpet so far, we also want to be able to deal with noise. So here, well, there are sufficient cases where B, C, and D happen together, but there might even be a case if we make B, C, D, let's say, wide enough, right? B, C, D, E, F, G, blah, 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 that we never really see each of those really together. There's always one missing, but under the condition of the left-hand side of the rule, it's a very dense block, right? That can't be explained otherwise. So this is basically what we would like to have. Is that clear? It's also clear that we can't do this with the tools we had last week. That's important, right? Otherwise, I'm just wasting your time. Now, we're going to be using MDL again, right? I promise you at one moment in this lecture, we're not going to be using MDL, but today is not the day. Actually, today is the day, but later in the lecture. But at 2/3, roughly. You already know what MDL is, so I don't need to spend so much time. Right? But, the idea here is we have some data, like on the right-hand side, and the model is now a set of rules rather than a set of patterns. Okay, conceptually not so different. And we want to now find that rule set, that set of rules that minimizes the total number of bits. Okay, fine. We already saw last week that life is difficult, right? Just if you want to find patterns, this is exponential, pattern sets, doubly exponential. But for rules, well, this second 2 turns into a 3, which does not make life, oh, oops, better. What are you doing? Just like last week, I mean, in this case, we could actually also prove it. It's not submodular, it's not monotone. So yeah, there is really no structure we can exploit, so we resort to heuristics. I already told you this is a pattern in our papers, so here it is, even in the presentation. So what did we come up with as an algorithm to optimize the score? I'm not going to go into the details of the score. This is not so important right now. For that, you can read the paper. So the main idea is that we start with some data. And in step 0, we initialize our rule set with the singleton or empty rules. We have different names for this, which are all of the kind empty head to A. This allows us to encode any data set over this alphabet, right? No matter how they're distributed, we can encode everything. We also keep our hands off those. We never kick them out, right? This is another thing that last week I forgot to say. Also in crimp and slim, the singletons we always keep for the same reason. We don't want to, well, be stuck in a corner like, oh, if only we had A, we could have encoded this, now I don't. That would be stupid. So they're always there. And we don't penalize the model for that. This is how we start. Good. So that's the initialization. Oops. Bloop. Then, much in the idea of slim, what we now start to do is we take the current model and we start to consider combinations of existing rules as candidates to be added to the model. So a greedy bottom-up strategy. For example, we have empty to A, we have empty to B. We can merge this into empty to AB. But we can also merge it to A to B, basically chaining them up, right? Empty to A to B, right? I mean, that, and also the other way around and whatnot, right? So we get a quite large number, I mean, roughly quadratic. That is our candidate set. Bloop. Okay, now, do we want to test all of those? No, because and definitely not in every iteration, right? I mean, that. So what we want to do is rather than, and why don't we want to do this? Because for every candidate, we have to make a pass over the data and cover every record, which is very costly, right? So what we can do instead is use the information we already have of how well the old model was functioning and how often we saw an empty to A and so on and so forth, to build an estimate of how often we think empty to AB will be used. And if we know how often it will be used and, well, how empty to A and empty to B will be affected, we can estimate what would be the gain overall. And we can do this for everything, not just empty to AB, of course, right? So we came up with an estimate, and, well, I'm not sure. I mean, does anybody have good enough eyesight to see that it actually is an estimate and not an exact calculation? Because it is almost an exact calculation. When Jonas and I were defining this, and then he came back with this plot, I basically told him, okay, please redo it. I mean, please re-implement this because I don't really trust it. Because the moment that the estimate versus the, so this is the estimated gain, this is the actual gain. If this is really purely a diagonal, then we seem to have an exact calculation, which should not be possible because what? Over here, there, you see it? This one dot, right? I mean, that is the one that convinced me. It is actually an estimate. I mean, I checked the code. It's not that for 1 point, it's just an offset. No, that sometimes it goes a little bit wrong, but we can estimate this very, very well. And it allows us to kick out all candidates for which we are certain that, or certain, almost certain, that they're not going to give us a gain in bits, but they're only going to cost us more bits, right? Those are useless. It also gives us something more. So this is what we do over here. It also gives us the opportunity to focus on those candidates that we think will give us the most gain. And then that allows us to define a strategy that if we now take the one that we think gives us the most gain and it gives us gain at all or a good amount of gain, let's forget about all the others. We can be super greedy in that sense, which speeds up things massively. So that's what we do. Yeah, okay, well actually this I'm just going to quickly skip because this is more like how do we cover the data. We always start with the biggest rules, similarly as in crimp and slim, right? We want to cover within as few as possible bits, so use as few as possible rules that are as long as possible. Avoid singletons, and wherever you need, use the singletons, and there you are. So, score both of the current model and the previous model, see which one is best, take it, rinse and repeat. Very simple. And you repeat this until convergence. And, well, we have a proof that our score is not submodular. But if I would have shown you this plot and I would have asked, okay, which behavior do we see here? I hope that at least some of you would have said, okay, well, that looks like submodular, right? There is a law of diminishing returns going on here. At the beginning, you get very big gains and then become smaller and smaller and smaller. Well, we can prove this is not true, but we can show that, well, it looks pretty darn like that, right? That, of course, by the algorithm taking always the best one, you kind of force this behavior. So who knows, if you would evaluate exhaustively, there might be actual dips, right? I mean, that is possible, but unlikely. This is to illustrate that even though this compression gain has then, well, let's say theoretical properties that are perhaps not so amenable, like not submodular, not monotone, on paper, in practice, it kind of does. So this is where theory and practice sort of bite each other, and where I think it's very good to know the theory such that you can also, well, in practice, almost ignore it, right? Good. Let's show you if this works. You'll be reading a lot of papers, both in this course and in other courses, I mean, in other times of your lifetime as well. But people try to get their papers past reviewers. That is the main goal, right? I mean, that people have, which is sad because the main goal should be communicating science, right? Showing insights. I'm not saying that I'm better than other people, right? I also want our papers to get accepted, but I try to play the game as fair as I can. And one of the experiments I find really important, especially for these unsupervised tasks we have here, is to figure out what happens if the data contains nothing, if there is no structure to be discovered. I want to have a sanity check. And when I say it like this, I mean, probably like, yeah, of course, right? That's, but the number of papers that actually provide you a sanity check is remarkably small. And if you think about it, this is perhaps even more important to give the sanity check than the, look, on this data I found this, or I have that and that score, because that is great on that data set, but what if there's nothing to be found? For example, I was very happy when, this is a few years ago, when people showed that if you take a deep learning architecture with sufficient capacity, and you take a certain data set, and you're like, oh my God, I get 99.9% accuracy, yay. Okay, what if we now completely scramble the labels? There is no structure anymore. If you train it new, it can still get 99.9% accuracy. or something very close to it, which is on the one hand super impressive, but also very, very scary, right? This means that you're wielding a very sharp blade and you really need to be really careful that you don't cut off your own ears or nose, right? Yes, Mikael. Yes, yeah, exactly. And preventing your deep net from overfitting, yeah, well, good luck with that, right? So what this shows is that you need to be really careful with this. And there are techniques to figure out how badly am I overfitting or not, right? But you need to be aware of this. This is, again, where theory and practice, right? I mean, they go kind of hand in hand. And just going pure practice, not a great thing, right? Pure theory, I mean, that's useless. You have to balance those two. Okay, now let's see. Pure noise. Two methods. Kingfisher is the statistics-based pattern miner, only reports patterns that are statistically significantly more frequent than you would expect. Well, this data is completely random, completely like sampled uniformly at random, right? And the only thing we adjust is the number of ones in the data, right? So basically we draw certain metrics, and every cell in the matrix is a Bernoulli variable, right? Where we just change the probability. That's the x-axis, right? I mean, not. It should be here, should be at 0, but we are here, which is a little bit scary. Then hyper, that's a method that finds big blocks. It likes big blocks and it cannot lie. And here at the bottom, the animation is borked beyond belief, which is strange because I fixed this yesterday. PowerPoint is a gift that keeps on giving. Here at the bottom, we have two methods, namely pack and grab, that I would say because of the compression idea, like if there's only noise, compression is not going to allow anything into the model because it's not going to give any gain, right? You can just describe the data as well with the independence model as with a more complicated one. So those actually find the correct answer, nothing, which is good. That is convincing, at least if there's nothing, the chances of overfitting are at least not as big. Now here we have data in which we do plant patterns, right? We plant certain blocks, plant certain rules, and these are the number of rules that are being recovered. We see that pack sort of shoots through the roof, which is basically if you are using a method for something it's really not meant to be doing, then it's not going to work well. I mean, yeah, surprise, surprise, right? The others, Kingfisher and Hyper, you can see they, well, find a few thousands of rules. Now, grab is over here. And perhaps you can see it. There is this dashed line, black, pretty much at the same height as grab. That is the ground truth number of patterns. So this doesn't show that grab finds the correct patterns, at least not yet, right? I mean, actually, those results I'm not going to show today. It does, but it finds the right number. It's not going to overwhelm you. Right? So again, good. I removed some results because I wanted to save a bit of time, but I am going to show you, again, the results on the abstract data that we already saw last week for a different method. And why do I show them again? Because, well, I can show that grab finds patterns very similar. We have here, well, both. This is JMLR, this is Machine Learning, Journal of Machine Learning Research, or really machine learning papers. And the ICDM is the International Conference for Data Mining, more data mining, more frequent pattern mining. I mean, there's also some SVM stuff there going on. We saw that last week. The patterns, well, they make sense. And these are the rules that we find. And this is interesting. Because here, in a machine learning paper, when we talk about support vector machine, well, it makes sense to have this as a pattern, right? I mean, this support vector machine increases the probability of seeing SVM, which also makes sense because often you write support vector machine and between bracket SVM, right? And then afterwards maybe use SVM or not, right? That, same thing with kernel space. Kernel space, whoops. What is? Okay, this is a trick I've never done before. Okay, kernel space is both a pattern, because well, SVMs work in kernel space, increases the probability of seeing reproducing Hilbert kernel space, which makes sense because you're not write the entire thing all the time, right? But, so. Also note that the intuition that we have here with rules is very different than a logical implication, right? The logical implication would be the other way around. And we're finding, well, the illogical implication, you could say. This confused the audience at first very much because there were lots of people who worked on logics. And you're like, okay, if you have an error, and you're doing it wrong. Like, no, we're not. I mean, that's it. Yeah, but then we had to explain, okay, but if you do it like this, you don't find anything interesting. And like, they were kind of shocked. And then like, okay, I'm not sure if I want to live in this universe. But so here we have support vector machine to SVM. In the data mining papers, we have support towards vector machine SVM. Does this make sense? Yes. Last week we learned that for frequent patterns, we're talking about frequency, but also for support. So the chance of seeing support is much higher. And then, well, vector machine is basically a subpattern of that. So for the left-hand side, we now know that, well, support is not an overloaded term. For the right-hand side, it is. So it finds differences already. Good. So just to wrap up, it was not just done in 10, 20 minutes back in the day, but we now have a method that can find rules from data as well as patterns. More expressive, and it allows us to get more insight into data, to also be better at finding anomalies, to be better at clustering, and, right? So in the end, this is a very nice step forward. Good. Any questions about this part specifically? No? Still, Timon. Yes, that's a very good question. Our interpretation in the end was a conditional probability. So the conditional, if I have a pattern A, B, C, right, let me just go to the example that I hear at the beginning. Well, no, don't go here. Up here. So here, I would say this is a pattern. Also in the interpretation as we had it last week. This is really A and B and C, right? That there it is conjunctive, like they really belong together, and it's essentially doesn't make sense to pluck them apart. You could say, okay, this is A implies B and C, but also C implies, it doesn't matter. And then we need a new type of word rather than putting us as a rule, right? It's in this direction, that direction. No, we have the word, this is, let's say, a family of rules, and we call that a pattern. That is our interpretation. Whereas here, for example, here, this doesn't really make so much sense. If I say, okay, this is A and B and C, well, then there is a huge amount of error at the AB. There's no C, and B, so that it's not really A, B, C, right? I mean, sure, A, B, C together have some interaction, but what kind of interaction, that is then of interest. And B and C are kind of independent there, right? Just mean, call it, well, both conditionally on A and in general. But A and B are not independent, and A and C are also not independent. But it's not that we can just flip the arrow from A to B to A, just like that. We have really a different statement. So that makes this a rule. So the probability of B is increased in the context of A, and the probability of C is increased in the context of A, which is different than the probability of B and C is increased in the context of A. Yeah, sure. In a way we can. So this one, yeah, but we want to be more compact. That is the idea. So this one on the left, we could say this is A and B and B and C, and A and C. And I can transmit exactly the same information, the same data. The question is, does this now take me more effort? And yeah, now I need 3 special words, and it's just a simple example, rather than just 2, right? And there can be cases where that is actually true, where I need these 3 patterns, but not here. Here, I can be more compact by saying it's A to B and A to C. Yep, good. Oops. Okay, so now you've seen now a number of these pattern mining methods, and I can show you a lot more. Throughout the course, we'll see 1 or 2 more, I think. But let's now also see a little bit like what can we do with it. And I've been bashing a little bit about on neural networks, right? I love to do that. I mean, I also love them for when they're good, but I mean, I don't like being just on the hype train just for that. But one of the questions we asked a number of years ago is, well, what's in the box? So everybody's always talking about neural networks being a black box, right? I mean, that's and we need to open up the box and we need to look inside the box and whatnot. And well, we agree that that's a very noble goal. That's a very smart thing to do. And then we looked a little bit about what are people doing, and we were not very happy with it. And well, as the saying goes, if all you have is a hammer, every problem looks like a nail. This is not necessarily what we were trying to do. We didn't set out like, okay, we're going to solve this with pattern mining, but we stumbled upon a way how we can use grab, or if you wish, also slim and crimp perhaps, but that's not what we did, in a way that gives us a lot of insight into neural networks that, at least then, nobody else could do. And I'm going to show you how. So these are, by the way, the slides from, this was the master thesis of Anna. The paper in the end, so she did not continue a PhD because of personal reasons. So Jonas is the first author of the paper, but I'm using the slides of her master thesis representation, right? So also to give you a bit of an insight how you can do it. I have to say Anna is brilliant, right? Both in her work as well as presentation skills. So if you're like, wow, okay, I'm not sure if I can manage this, well then aspire to it at least. What's in the box? I guess that everybody knows what a neural network is. Is there anybody who doesn't know what a neural network is? Good. Then either you're super shy or you do know. This is what the toy version that we're going to be looking at. Specifically, we're also going to be thinking not just about feed-forward neural networks, but at convolutional feed-forward neural networks, right? In this presentation, I mean, so the paper was published in 2021, and we worked on it roughly in 2020. I mean, it's not that transformers did not exist attention as mechanisms, but we didn't focus on it, right? Okay, what is the general idea? We have some input, we have filters, these are the convolutional layers, right? And then afterwards, the output of those then, either with a max pool or whatnot, then you have your regular neural network. Right? And in practice, what we get is that we have some input, we have a black box, and the black box gives you then, okay, well, this image is the third thingy, right? I mean, whatever you train it to be. Could be regression, could be classification. So we want to explain, why do we care? Well, especially if we look into medical applications. Does this person have, let's say, a brain tumor, yes or no, right? I mean, yeah, okay. Just yes and no by itself is not enough. Why are you saying what you're saying? Self-driving, right? Should we stop? Should we go? Right? Why? That's and just mean in general. So what is out there? Like I said, this is a few years dated, and the field is fast moving, right? If you're not like, my favorite method is not there, yeah, bear with me, right? I mean, Anna did not have a crystal ball, huh? No. So what is out there? There's model distillation. I mean, model distillation is still one of the preferred techniques. I mean, that you could say God knows why, but what is the idea? We have now a neural network. We feed it a lot of inputs, and then we have the outputs the network gives. And we just say, we don't know what the neural network is doing. What could we do? Well, we remove the neural network, and we replace it with another model that we can interpret, or we say that we can interpret. We could learn a decision tree. We now learn a decision tree between the input and the outputs of the neural network. And that, we say, is the explanation of what the neural network is doing. That does not make any sense. Just imagine that we have the perfect neural network that does everything perfectly. It might be completely overfit, right? And so on the training data, right? I mean, it may actually be who says that the decision tree can actually mimic this behavior? We don't know, right? And that's, of course, we can train a decision tree that also gets perfect scores if we just make it pushy enough, right? I mean, there is no connection between what the decision tree is doing and the neural network is doing, unless we are extremely lucky and that the neural network is actually mimicking a decision tree. But why would it? Huh? So, yeah, it doesn't make sense. Also, anybody who tells you that a decision tree is explainable or interpretable, you should at least mentally slap them around a little bit, unless they make the additional comment, as long as the tree is relatively shallow. The moment you have a deep tree with all these conditional steps, right? Each of these steps are basically conditional probabilities. No person can keep this all in their mind. None, right? This is nonsense. So yeah. And random forests, right, I mean, are even worse in a sense, because then you have many decision trees and you use all of them at the same time, and yeah. Work great if you want to have good prediction out of the box, but if you want explainability, no. Good, what is out there otherwise? There is prototyping and saliency maps. So let's look first into prototypes. Prototypes is the idea that we train the neural network, we lay out, and we say, ooh, what does that neuron do? The green one over there, in the top right. And what we do is we use effectively backpropagation in reverse, right? So we propagate, I can't find the word. We basically try to generate that input to the network that activates that neuron most. Or in my way of phrasing it, that makes that neuron extremely happy, right? I mean, we're really trying to find the ideal input for that neuron. And well, there's different techniques that can do this. And then you get images like this. We'll see a few in a second, I mean, or in a few seconds. Yeah, well, it's not super visible because I didn't blow it up, but what can you do? And what can you see in this? Typically, nothing. We can also do this, however, for convolutional filters. And if we're lucky, then we can see, okay, well, this is a bit like a dog face type of thing, right? I mean, that's in a core, maybe, right? But you have to be a bit creative there. What about groups of neurons? Because neural networks, they tend to not encode things like here in a single neuron. They don't have a grandmother neuron as such, right? Just like in your brains, there's also not one single neuron that remembers who your grandmother is, which would be a bit shit if you have, well, one drink too many or you're a bad night of sleep and all of a sudden you don't know who grandma is. That would be a bit unfortunate, right? So grandma is stored in an area over multiple neurons. We see similar behavior in artificial neural networks, not so much because it really wants to hold on to the memory of grandma, but because we use techniques like dropout. In dropout, we basically, during training, randomly disable certain neurons, right, in every epoch, in every iteration, right? I mean, that's, which means that, yeah, the network is really pushed to store grandma both here and there, because it might well be that otherwise it doesn't remember grandma when it's being asked who is that, right? So basically we get that sets of neurons start to encode a concept. And in the ideal case, let's say grandma is stored over 3 neurons, right? All 3 are active, and then, oh, yeah, that's grandma. If one is being disabled, then with 2 neurons saying like, yeah, this is probably grandma, and one is like, yeah, well, could be grandma, right? Something like this. That is a bit of the idea there. There's one next step that is happening called superpositions and polysemanticity, that a single neuron does not necessarily encode, is not necessarily just part of one concept, let's say grandma, but can also be part in recognizing, let's say, fire truck, together with a few others. So there's not a single role to one neuron, right? There's really a many-to-many mapping. So you need to have something to deal with that. Which means that for this prototyping, we kind of would like to find, okay, these are the 3 grandma neurons. And if we visualize those 3, then we're going to get a picture of grandma, right? And otherwise, well, yeah, I don't know. What is the average between grandma and a fire truck? Because that's what we're going to get if we do this for a single neuron, right? And neural networks do this all the time. There's very few neurons that are monosemantic. So, okay, suppose that we now let's find these groups of neurons. You, of course, already have an advantage over the general audience, right? Because finding groups of neurons, that sounds like pattern mining. But it's a very different mind game than what you normally have in machine learning, right? Finding groups of things in combinatorial, don't want that. Now, one of the key arguments not to do this is that there's 2 to the power, however large your network is. This is many, right? This explodes, this is icky, do not want. And this is where the stuff that I've been explaining last week and just in the previous half an hour comes to play. Because we can use grab to find rules between the neuron activations of the final layer to the pre-final layer. And then, after we've done that, we can do this from the pre-final layer to the pre-pre-final layer. And that way we can hop our way through the network and find how the network propagates information. This is very abstract. I see some of you looking or frowning in a way that I wasn't sure it was physically possible. I'll explain you. This is generally the idea that we want to achieve. And then we can visualize very directly, like, okay, well, this, let's say this is grandma, and grandma, well, this is basically I don't know, grandma's hairdo, and these are grandma's glasses, right? And this is perhaps the background, right? The living room in which she typically sits, right? And most pictures the network have seen, are in that, if they see that particular room, it tends to be grandma. And then where in the filters do we expect it? Well, grandma's hair is over here, and so on and so forth. Now, how can we do this? We take 2 layers. In practice, we take 2 consecutive layers. It's not strictly necessary. This is really just to make things a bit more easy to handle for ourselves and a bit more interpretable. Let's give some labels. I mean, that's because what do we normally have? We have an image that we put in this case, a pug. And then the network says as output, yeah, this is a pug. It's not a Pekingese, it's not a Great Dane, it's not a Husky, and so on and so forth, right? This is the output after a softmax or whatnot, or a linear layer. The layers before, they tend to be, well, continuous valued, right? This is what neural networks, how they operate in between, because it's all linear algebra. So this neuron has an activation of 0.8, and these are all 0, and this is 0.16, right? Because our L1, L2 regularization has been doing its job. Now what we can do is what we can transform these activations into a table. So we have on the left-hand side the activations for layer 1, right? So the pre-final layer. And on the right-hand side, we have then the activations of the final layer, or of the other layer. In this case, the other layer, they're already binary, right? Because this is basically how the network works. That doesn't really matter so much. We do this for not just a single image of a pug. We do this for all images that we feel like running through the network, because we want to get a feel how does the network perceive the world, right? So in a way, the more images we give it, the better, because the better we're going to find the patterns that really make up this decision process. So we arrive at something like this. And so we have images of Pekingese, of Great Danes, of Huskies, and so on and so forth. Now, what do we do with this? This is where if all you have is a hammer, every problem looks like a nail, comes in perhaps a little bit. But I still would be an unfair characterization. We can binarize these activations. That is the idea. If we binarize them, then all of a sudden they will look something like this. And if we have binary data, we can use pattern mining. Why does binarization make sense? Well, in biological neural networks, you have a baseline activation of neurons. Basically, they're off, right? Or they start to spike very frequently, right? So activation has sort of like a sigmoidal type of shape. This is one of the reasons why artificial neural networks, for a long time, we used a tangent, or we use a sigmoidal or whatnot, right, as activation functions. Do we still do this? Not always. You could say that ReLUs and all that entire family are a bit more popular because they're easier to train. But also with the ReLU, you have the area where it's off, and then it's on. And then it can be more on or less on. Yeah, sure. We can handle that, right? But there is an off part, there's an on part. That we throw away the amplitude, fine. That's effectively what we do here. Is the neuron on for a certain input, or is it off? That's what we work with. We binarize the values, and then you could say we run grab. Basically, we run grab on steroids, because it needed to scale up, and it needed to do some other things. It needed to be able to do ORs, it needed to be XORs and whatnot. So Anna really deserves her 1.0, but that's another story. But we would like to find, and this is why we started from grab, are rules of the kind that, hey, pug or Pekingese, right? So this is really an OR structure, very much goes in this direction. We have Great Dane. Look, those are these two, really, right? Then Great Dane and Husky, or Husky could perhaps be this one, right? So big dog and small dog, maybe those are what the network is doing. I don't know, this is a made up example. But Husky, well, that goes to this one. So we want to find these activation rules. If I see Husky, right, and in the Husky neuron, I tend to see the other one active. So we want all rules. We already said, I mean, this is what grab can't do. How it technically works, it's mostly in the search. That's not so super exciting for now. We have these conjunctive patterns. We already know how that works. We obviously want to have noisy rules, right? Grab can already do that. We just had to, well, give it some steroids in that sense, and then we can find noisy rules. But with noisy rules, we also have another question that we need to answer. So here, these neurons, V1, 2, 3, 4, right? I mean, they have a very noisy relationship with U1. how many of those do we want to have that are on, right? So how many of these neurons do we want to see to be active before we say like, okay, now we would also expect U1 to be active. All 4? No, that's too restrictive. 3 out of 4? 2 out of 4? 1 out of 4? You don't want to give this parameter up front, right? This would be weird because how would you know this? And this will be different from the one rule than from the other, right? So how could we do this? Hey, we can use compression. We can simply consider the different variants of interpreting this one. So 3 or more, 2 or more, 1 or more, or just as an or, and then see what would be the activation vector that we get, right, for this part of the rule. Consider this then as a rule and see how well does it compress. And if it's a bad choice, MDL is not going to like it. So if we allow, let's say, at least one of these, well, then it will always be active. But that may not be good, right? Maybe we need to have at least 2 or at least 3. It depends very much on how these neurons behave. But by, again, using compression as a way to decide what is the best option to describe the data, because that's what we want to do, right? Those conditions that describe the data best. So this is what we're going to get here. In this case, at least 3 of these 4, then we see U1. Yeah, so grab with a few engineering tricks. This is underselling it, but for now, this is fine. I really want to more carry the idea across that we can use this rule mining, which, yeah, how different is this from pattern mining, into a new application domain. You know what MDL is, so I'm going to skip over this. The method, by the way, is called explain. I'm really happy with the name. This is a good name. Right? Algorithms should have a good name, right? So crimp was good, slim was good, grab is good, explain is good. Not all our methods have good names, but many, many do. So, okay, yeah, how do we find them? You just saw a grab, so I don't think we need to explain this part of the presentation, right? I mean, Anna did not have the luxury of an audience who had just seen this. So up, we merge rules, blah, 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 blah. So, good. So let's see. What can we do with this, right? And just to recap, we take a neural network, we run a number of samples through it, we record the activations, we binarize them, we take the activations of 2 consecutive layers, we run grab on steroids. That is the idea. We have results also on Cypher10 and a number of other, on MNIST. Those are a bit too analytical for today. So I focus on some results on ImageNet. As you know, ImageNet is a real world, a collection of real world images, right? I mean, small ones, I mean, not, and one of the standard benchmarks, right? I mean, if your method gets 99.97% accuracy, right, rather than 99.96, oh my God, your method is so much better because probably it's overfitting, but everybody just ignores that. That's what ImageNet is often used for. But because it's so often used for us, given that we just want to show what did your network now learn, we don't really care about that this benchmark is overused. We were using VCC12, I think. We took an off-the-shelf convolutional feed-forward neural network because we were not interested in playing the accuracy game. We just wanted to explain what is going on, right? So what we're looking at is a convolutional neural network with 5 convolutional layers and 2 fully connected flattened layers. Okay. First, we run grab from the output to the pre-final layer, just like I did in the example, right? But, no surprises there. What it found is that these 7 outputs, they tend to, well, share a lot of pre-processing, let's say a group of neurons in the layer before. So it found it out itself. Do we have a dog expert in the room? No? Does anybody already see, let's say, a pattern amongst these dog breed names? Okay. Okay. Yeah. Is that also what you wanted to say? Okay. Good. Good. Let's see. Let's see. So, so these 5 neurons, right? I mean, in the pre-final layer, they are strongly associated with these 7 labels, right? This is basically what it finds. Let's do prototyping, but let's do prototyping the old school way, right? So we visualize them one by one. We try to conjure up that input or an input that maximizes the output or the activation for each individually. We get images like this. You still can't see them, but now I'm going to blow them up. I don't know if they would have shown this in biology class, right, or something like this. Okay, maybe this is like cells merging, or I don't know, right? I mean, that's now that we know it was dog. Maybe it's just whiskers. Yeah, sure, right? No, stripies and circles, whatever it is, right? Next one. Butterflies, butterfly wings, maybe. Maybe it's small dogs with big whiskers and wings, right? Maybe, who knows? Faces. If you would twist my arm, it's again butterflies or perhaps cat faces. If I would not know the labels, right? I mean, if I know the labels, then maybe here you see like a dog snout, something like this with eyes. I don't know, but we're reading a lot in the tea leaves here, right? I mean, that's this one. This I would say is cats. These are cats, right? I mean, but these labels had nothing to do with cats. But had I shown you this, would have asked you cats. Maybe next year I'm going to flip this around. First show you the images, then you tell me what you see, and then I tell you that you're wrong. I would say this is frogs. Yeah, a frog and 4 birds. I mean, that is, I'm fine. I'm fine with that. That's yeah. Well, that is basically the status of prototyping, right? But where we found it, and you see this problem of this polysymmetricity, right? I mean that. So, if we now take these 5 images together, or the 5 neurons together, and we prototype those 5, we get this. And it is still a bit nightmarish, right? But and here is this one of those small, we call this the ugly dog pattern, right? So not just small, but it's like a type of dogs. Here you have a face of one, here you have a face of another, and you have here also this maximum activation, right? One of the key problems is that it really tries to come up with an image that maximally activates that neuron rather than a typical image that activates this neuron, right? So you're going to get these weird things, but this is more interpretable, I would say, than these individual ones, right? At least we're not talking about butterflies anymore. If you get nightmares of this, I'm sorry, I apologize, but yeah, well, should have given a trigger warning. We can do this for other, well, so some other results that we found, right? And that, for example, here on the left-hand side, the red fox and the lesser panda. So I think everybody knows what a red fox or a fox, regular fox, looks like, right? I mean, that's like the small panda is this reddish panda with this fluffy ears. Yeah, it's basically, does it have reddish furry ears? That is what this is encoding, right? So that's what these 2 neurons encode, and hence they're relevant for both these labels. The middle one, leopard, snow leopard, and cheetah. Yeah, spotty pattern, spotty fur. Give it a name, right? I mean, okay, nice. And on the right, and these are 7 neurons, by the way. So just, it's really like, it loves spots, right? I mean, those of you who did the assignment on deep learning, right? Beware leopard sova, there you have it. On the right-hand side, jackfruit and squirrel monkey. It sounds a bit weird, right? I don't have the images of jackfruit and squirrel monkey to pull up. Let's say for the interested audience member, look it up after the lecture. Because this pattern, we were also like, okay, wait, what? Stupid method? No, not stupid method. It makes a lot of sense. It makes a lot of sense. Good. What we can also do is, well, figure out if there are subclasses of a certain class, right? Or of a certain group of classes. For example, we found for label Great Dane that there's not just one pattern or one rule, right? But there's actually 3 rules, which also surprised us a little bit because, well, why would you do this? Why don't you combine them? Well, compression should have a reason to do this, right? Otherwise it would have to merge this and has the option to do so. So what we found, if we prototype those 3 patterns, then we get this one, and this one. I'm not saying that this is maximally informative, but what we could do rather than just prototyping like we did, we can also go through the input images and see which images activate now specifically those neurons. And then we found this. There are Great Danes which are very brownish. there are, well, this is the side dog pattern. I mean, this is really the triggers on dogs on the side. This is the dog face pattern. So you see it looks a bit like a hippo if you blow it up, but really these eyes and nose, you see it, but not, and you have Great Danes that are spotty. And it really uses the network 3 different pathways to recognize different types of Great Dane. And in the end says, okay, yeah, well, any of those you want me to say Great Dane, I will say Great Dane for you. Which is kind of cute, right? So it's not 1 pathway, but rather 3. Which for Great Danes, okay, well, potato, I don't care so much. But if we were now to recognize, let's go back to the original motivating example, X-ray scans or whatnot, right, of brain tumors or other type of medical cases, just saying, hey, yes, it's cancer. It's good if it can do this with high accuracy. But if we can learn something from why is it saying what, and are there subtypes, or are there maybe, well, certain classes where it's cheating, right? I gave the example with CatFit, right? I mean, that there's, well, labels on the input images which it may be looking at. That might be that for some images, I see a label, I just say that it's COVID. And for others, I'm actually analyzing it. Well, then we can also actively intervene on the network and tell it, don't do this, we just turn off those neurons, or we continue training, fine-tuning, where we really penalize the activation of certain sets of neurons, and this way, well, steer or align the network. It's pretty neat. One of these two images is concerned with the dog breed Husky, and one is concerned with the dog breed Malamute. Which one is which? Which one is the Malamute? This? Yeah? Or that one is the Malamute. So you're saying this is the Malamute, and you're saying this is the Malamute. Great. Okay, because then we have that. Why? I first ask you and then you. Mm-hmm. Okay. Good, yeah, fair. What do you say? Mm-hmm. Yeah. Yeah, okay, yeah, both make sense. Okay, I didn't trick you. It's not like, well, neither. Ha ha ha. They're both Pekingese. No. Okay, this is indeed the Malamute, and that is the Husky. You were right that this one is more fluffy. That is indeed the key, let's say, differentiator. Not because they live in more cold climates, because Huskies, they're being used by Eskimos and whatnot, right? I mean, Inuit, I should say, of course. Also in polar regions, they love the cold, right? I mean, you can't make a Husky more happy than putting it into a freezer, right? I mean, just with enough air, of course. I mean, Malamutes are, Malamutes are actually used to scare off bears. They hunt bears. But this is why they're used. I mean, Huskies are used to help with sledding. Okay, good. Now we know what to look for, right? I mean, I didn't before the example was given to me, and I also had it wrong because, like, well, a Husky is like a black and whitish dog, right? I mean, it's both Huskies. I'll just choose one. But we can ask now, neural network, okay, which neurons do you use to classify Husky? Which ones do you use to classify Malamute? And we can look at the, well, which neuron do we use for both, and which are the complements? And we can prototype, well, in different sets, right? So that can give us some insight into what might it be looking for. And there, I mean, I didn't blow these up, but for the Malamute, we have this. Jointly, we have that, which sort of becomes a bit of like a Husky-ish face, right? I mean, not, again, is it super clear? I don't know. Here we have a bit more sharply drawn face. This one is more fluffy. I mean, we're reading a lot in the tea leaves, right? I mean, because prototyping is imperfect and we're just using this as a tool that is there, right? I mean, Jonas, meanwhile, has been working on better prototyping, right? So to also show that this actually is doing really good stuff. But indeed, it finds that Malamutes, specifically, more fluffy, Huskies more sharply drawn, but in general, well, both have this black whitish face. That's a little bit, let's say, the reasoning that is going on here. It's just neat. Now we've been looking primarily at the last 2 layers. And I've been promising you that, hey, but we can also chain this. What can we do there? For example, for the label totem pole, it finds a set of, I think, 7 neurons at the top of my head. I don't know exactly, but that doesn't really matter. And we can prototype them. And this looks quite totem polish in colors, and it's like a cylindrical shape, and there's some wingy thing. It's not perfect, but yeah, right? I mean, that's... what we can now do is we can now, rather than considering the last layer and the pre-final layer, we can consider the pre-final layer and the pre-pre-final layer, right? And there's different ways on how we can connect those things, right? I mean, we can just mine them verbatim. We can use the activations of the patterns that we found, for example, for this one, these 7 neurons, for which records are they on, respectively off, and that give that already to grab as input for the one layer, right? And well, you have different advantages and disadvantages for all setups. So let's go ahead. What do we find? We find that in the pre-pre-final layer, another set of neurons that is very strongly associated with the pre-final one. And if we prototype this, well, we still get this longitudinal shape, and we get sort of like more animal head, some like a fish mouth or something like this. Okay, well, we can go one step further, then we're already at the convolutional layers. And for each of those neurons, they then start to correspond to filters, we can see where do they tend to be active. And if we prototype that part specifically, what do we get? And we see here at the top, this one is active really at the top. We get sort of like, well, 2 eyes. Well, at totem poles, you often have some animal or human head at the top, right? I mean, that's we have some crown type of thing, right? Or some let's say pagoda type roof. That makes sense. It's symmetric and round. Well, 2 sides. I don't really know how to interpret this one. And this is also, I would say, the hat or something like this. But the key point is not so much what do we see, but we can now really focus on where is it expecting certain inputs. This is the typical way how it processes information for a totem pole. And rather than using prototypes, we can do exactly the same thing as with the Great Danes. We can also look for those images that maximally activated. Of course, both have their advantages and disadvantages again, because looking at the images that maximally activated, it might not show us the typical behavior, but it shows us the behavior on one specific image. Whereas the prototyping does show us the typical behavior, but yeah, it's a very fuzzy lens, right? So this is where there's a lot of room in the middle where interesting things can be thought of. But now all of a sudden we went from pattern mining, from mining patterns of the kind beer and diapers, to gaining insights into neural networks. What I'm not showing you here is that if we intervene on any of these sets that we find, basically by turning them off or making them random, then the performance for the class that we associated with it, right, or how we found it, this crashes. And really much more than if we do this by random. So we really have the information flow in the network by its tail. That is really cool. If we could visualize better, that would be great. Is grab the best way to do this, or explain in this particular case? No. It can't do these mutual exclusivities, and the noises are also... It ignores completely the neuron activations rather than on and off, so we throw away information. It's a combinatorial algorithm, so it scales quite reasonably with the heuristics we gave it, but can we make it search over the entire network in one go? Well, yes, if you want the result long after you're dead. Then it's fine, right? But otherwise, no. So there's plenty of steps that we can take on how to improve this. One thing how we actually apply, or how we explain, is that so Anton, who last week was sitting there, and he also handed something in, but maybe it was a bit too late for him then to also still join the lecture. So hello, Anton, if you're looking this online. He did his bachelor's thesis in my group. And we were looking at, can we detect, using explain, or a variant thereof, whether a neural network is biased in how it processes certain labels, in the sense that in predicting, let's say, doctor, right, does it have different pathways, and does it have different, let's say, preferences for, let's say, male doctors versus female doctors? Because it could be, right, that for example, there's the famous or infamous example where I think if you translate from Spanish to English or vice versa, that essentially every woman doctor gets turned into a nurse. That's, yeah, a bit unfortunate, to put it very mildly. I mean, that you would like to tell the neural network, don't do that, right? Pay extra attention here. Now, of course, this is an example that you will stumble upon quite directly, but what are the other biases? Which classes does it inherently prefer over others where we know that it shouldn't? And how can we pick this up? Well, we can do this through the lens of explain. So I like explain a lot, but the downsides are already discussed. One of the other things is that we are looking directly at the neuron activations and this polysymmetricity and this, well, let's just focus on that for now. The polysymmetricity, that multiple neurons are involved, right? We can kind of see through this if they are often enough co-activated, but it's not really perfect for that. So there's other techniques how we can abstract away from the actual neural activations and get another representation space which is cleaner, that really looks more at concepts. I mean, sparse autoencoders are one such tool. And then all of a sudden you can start thinking, hey, can we now do something like this, but that not directly to neural activation, but at that level? How do you make this work? Because those dimensions typically scale up massively, so we need a method that can really scale up from this. Lots of interesting research to be done. That topic, coincidentally, is the topic of TADA, who's writing a master thesis right now, and she's at least as smart as Anna, so maybe next year I'll have a complete new presentation for explain, but that comes too early for you. Yes? No, afterwards. So you give me a trained neural network and some set of inputs, which are somewhat representative for how you would like to use the neural network. It could be your training data, it could be your test data, it could be both. I don't care, right? Because I'm not going to judge you in terms of accuracy. The goal is what are, let's say, the typical information processing patterns inside the neural network for these inputs. That is the idea. Yes, exactly, yeah. Yeah, exactly. Well, this is all superpositions, polysymmetricity. These are all different sides of the same metal. But so these superpositions is really that one neuron can be involved in multiple, so grandma and fire truck. And so I didn't show you an explicit example for explain, but explain doesn't care if we have a pattern, let's say ABC and CDE. And ABC could be grandma, and or ABC is grandma, CDE is fire truck. That's absolutely fine, right? It can find that. So in that sense, we can deal with superpositions. And polysymmetricity you've already seen, right? So that you have multiple neurons. So when we wrote this paper, nobody even knew the word superposition or polysymmetricity, right? I mean, everybody was looking at these individual neurons and okay, a neuron has one meaning. They were stuck in that. So I'm not going to be that grandiose and arrogant. Okay, we solved the problem before it even arose. No, right? I mean, but we came up with a solution that already does this. And right now, every paper in explainability is like, oh my God, we can deal with either superposition or polysymmetricity. And we're so great. And like, yeah, they might be much better than what we did, right? Because progress advances very quickly. But yeah. And the neat thing is that we get these benefits by just approaching this problem slightly differently. By starting from the point like groups of neurons and allowing these to overlap and whatnot, right? Because really the explain as an algorithm is really not so far away from grab. So that is pretty neat. Good. You again? If easy is it would be easy, then well, if Tim Bauerschmidt would be in the room right now, he would start to cry probably. He wrote also his bachelor's thesis with me, and we have a variant of explain for transformers, but we actually stumbled upon a few other problems, namely in order to show, so if we can find which neurons now are co-active, right? For images, we can do this prototyping. How do we do this for text? And transformers typically are used for text if we ignore vision transformers. So we actually got sidetracked with developing essentially a prototyping technique to show that the neurons we found using his variant of explain actually do something meaningful. That was the one thing that we had to solve. And second, so in a convolutional feed-forward neural network, we can do this binarization. It's quite natural, on or off, right? And you can argue, okay, well, maybe in a ReLU, if we have this, right? So this is, so maybe on doesn't start here exactly, but maybe it starts here, right? And maybe we could say, you know what, we have off, we have a little bit on, and this is the very on, we have ternary data. This is all, I mean, we can do this, right? But in an attention mechanism, you kind of lose this interpretation because it's just, well, some linear algebra and out comes some values. And how do you binarize, let's say, the key, the value, or the query matrices? And we had some good ideas that work-ish, but I would say easy is not the word I would use. And there is still definitely a lot of progress to be made. But yeah, this is super interesting, and I would love to work more on that. But I only have 2 hands and 24 hours per day, so I can only do so much by myself. Good. Okay, any further questions? No? Okay, is everybody ready for the third part? Yeah? Because now we went from pattern mining, right, or rule mining to be precise, pattern mining, to applying rule mining onto neural networks. But what we can also do is use neural networks for, well, not rule mining just yet, but for pattern mining. Can we get away with the combinatorial nature of these algorithms? And can we apply them on truly high-dimensional data? Because one of the motivating examples I've been using are these genetic sequences. Then we're talking about hundreds of thousands of features. Well, if you just quickly do the math in your head, if you just, in the algorithms we've been looking at, we combine 2 existing patterns into a new one. That means if we have 100,000 features, we need to do 100,000 squared. And for each of those, we have to go over the data, or at least estimate. Do not want, right? That's really gets hairy very, very quickly. So can we use the neural network magic, which don't care so much about the dimensionality of the input space, to find us the patterns, well, in an automatic manner? Which is a bit related to what we saw previously, because there we wanted to get insight from a neural network that was already, well, trained. So now the question is, can we train a neural network that we know where to look and how to interpret what neurons do, such that we know, this is pattern ABC, this is DEF, and so on and so forth. That is the key idea of this final part. And what we will see is that the answer is yes, we can do this. And we get a method. It's not perfect by far, but it's neat. And it scales like a mother F. It scales really, really well. Which then raises the question, can we combine this with explain? I already gave away the answer. I mean, I'm currently working on it. TADA is doing great work there. That is the idea on how we can now level up in one direction. If you ask, okay, can we do this for, I'm going to explain it how to do this for patterns. Can we do this for rules? Yes. Lewis just finished his master thesis, and he developed rule maps. Where this name comes from, we will see in a second. Motivation. So again, these are the presentation slides from when Jonas presented this at KDD, which is the top conference for data mining, basically the NeurIPS of data mining. Hey, single nucleotide polymorphisms and samples. We already know this, right? We know where we want to find patterns. Oops. Supermarket data, we also know this. We would like to find that, well, in his case, beer and chicken. He didn't want to use the beer and diapers, or he didn't have an icon for diapers he wanted to use. That's perhaps also that. Traditional pattern mining methods deal well. So really the traditional, the old ones, right? They deal well with many, many samples. These are proposals that come from the database community, and they're millions or billions of records. They don't. That's billions is when things start to get interesting. Below that, they don't even get out of bed, right? So those algorithms, they scale really well. However, they explode in the terms of output, right? Then these compression-based methods, or these other pattern set mining methods, they scale much less well in the number of samples because we need to make passes over the data, right? Something's got to give. They curb the explosion. Neither are really good in terms of data that is very wide, that has very many features. The pattern set mining ones, they have to look at every combination, right? The traditional ones, there we can use some tricks, but the problem there is that, well, the output is going to explode even more. So you don't really even want to consider doing that. Meaning that pattern mining is a great idea in theory for these biological applications. And not just those, right? I mean, another example I can actually give here, which I should add. Let's make these not transactions, but let's make these programs, Android programs, or Apple, whatever. I mean, iPhone, right? Doesn't really matter. I mean, and then the columns are certain API calls or other software features. And what we would now like to find, are there any patterns that we can use to detect malware? Which, well, Google and the likes are doing all the time, but this is a bit of an arms race. So, and you can only train a classifier after, well, you know which samples to overfocus and fine tune on, right? So getting some insight, okay, wait a second, this is a cluster that of malware that's really abusing this set of API calls in a slightly different way, and this is now obfuscated. It's super useful. Now, this type of data tends to have hundreds of thousands up to millions of features. And they're all binary, so it's the perfect application for pattern mining, except that it's also the worst application of pattern mining, and it's basically the worst application for kind of anything, right? So this is another reason why we would like to have something that scales really well. Now, you already know that frequent pattern mining by itself sucks. You know that pattern set mining is kind of the solution to curb this explosion, but you don't want to do this in a combinatorial way. So these are some of the approaches. I mean, we have seen primarily this, maximum entropy, another notion from information theory. We have also, and others have also used extensively. Boolean matrix factorization is, well, matrix factorization of Boolean matrices. Essentially what you get, just like in regular matrix factorization, you get a factor matrix where you can interpret the column vector and the row vector, as the column vector is like which of the features do we expect to be on, and the row vector is like where in the data do we see this. So effectively, this is the pattern, and this is the where do we use the pattern. So matrix factorization, in a way, can be interpreted as a form of pattern mining. It's not exactly the same, right, depending on how you hold it, but still. Constraint-based pattern mining, let's ignore that for now. We already know that this is a very hairy problem, so heuristics, meh. What do we want? We want scalability. We want noise robustness, like we had in Grab, or even better. And ideally, well, no heuristics. We can't get all of those, sadly. What are we going to do? We're going to use a neural network. Does anybody have an idea how we could use a neural network? If we want to, so I give you some data, right? This transaction data, be it supermarket or whatnot, right? And say, okay, well, here's the data. Now train a neural network in whichever way you want, right? There's no labels. There's no labels. Train it whichever way you want, whatever neural network you want. My only demand is that after you've trained it, I want to be able, that you can tell me in your data we have patterns A, B, C, D, E, F, and so on and so forth. What would be a starting point? Does anybody have an idea? And maybe you can't fulfill all my wishes, right? So maybe you have a feeling for, hey, wait a second, if we take a step in that direction, maybe we can then figure something out. Yes? Mm-hmm. Possibly. Yeah. Okay. Self-supervised, of course, helps if we have unsupervised data, right? No labels. So then we have a network that can predict perhaps the patched out data very well, right? Sure, we can do. Self-supervision would be, okay, we take a transaction and we block certain parts out, or we scramble it a little bit, something like this, right? This is what you have in mind. We can do this, but that fulfills the unsupervised part, but not really the how can we read anything out part. And that's kind of important. Any other idea? Yeah? An autoencoder, yeah. Yes, that is what we're going to be doing. That's what we're going to be doing. But so for everybody, I mean, who might not have heard, we're going to be using an autoencoder. So effectively train a neural network that has to, well, give it input, has to reconstruct it. And we're going to constrain the middle layer. And we're going to be trying to do this such that we can interpret what is going on in the middle layer. And that is one of those sounds simple, but good luck trying to get it to work type of things, especially for binary data. I mean, neural networks don't like binary data. So what do? We start with an autoencoder. That's the key idea. Normally we train, so this is where the input comes in, X. Then we have an embedding layer. Sometimes there's multiple layers here if you have a deep autoencoder. We're not going to go there today. And then you have towards the output. The idea is that you want to learn those weights such that you can reconstruct X really well. So there's a very small difference between those two. This is already where having continuous valued input helps, because then for each of the individual features, you can say, okay, how close are you? You can use an L2 or whatever, some distance, right? I mean, that, for binary data, this already becomes kind of tricky, because for binary data, you either have it correct or you have it wrong. So it's a bit, the signal is very harsh, right? I mean, the contrast is very high. Let me actually first walk through the slides, I mean, not before I start to yabber too much. We have an encoder as the left-hand side, and we have a decoder as the right-hand side. The middle part is the embedding layer. Now, for embedded devices, like your phone and whatnot, using an autoencoder for detection or whatever, I mean, or to get a low rank representation in the embedding layer that can be sent then to a server to have not waste too much bandwidth, and then get the answer from the server for that. We need to have this easily computable. So what people have been doing is, rather than going for regular arbitrary weights, real valued weights, they've been truncating those, quantizing them. And what is the, well, let's say the most harsh quantization that you can do? Make it binary. The weight is either on or off. Harsh, but okay, right? Which of course makes the computation much easier. And if you have to do lots of multiplications with very high precision floating points, or just on or off, then it just becomes a summation, speeds things up, right? This is what people have been doing in embedded devices. So one of the ideas there is that we have then a binarized version of the weight matrix here, and same thing here. And then, well, we get also binary activations. Neurons are on and off, right? Everything is binary. This is already a very interesting idea by itself, because this works often remarkably well. So on the one hand, you want to have continuous valued weights to have a lot of freedom in your network, but if you constrain it like this, then often works really well already, if the network is sufficiently trained. The other idea is that if we want to train such a binarized network, that actually during the backward pass, we don't use binary weights. So effectively, we have continuous weights, matrices W1, W2 in the background, which when we want to propagate the errors, those are the ones that we use. But in the forward phase, we binarize those weights, and that's what we use in practice, right? Which makes sense, because if I were to not do this flip-floppy type of thing, then I might end up with a set of continuous valued weights that don't lend themselves to be binarized very well. And it's always a good idea to train a neural network in as similar a situation as you will be using it. So if you're going to be using it with binary weights, then well, let it get used to that situation. So this is an idea that we had, that we found in the literature on embedded devices. And these are not very new ideas, right? So in 1990, for example, I was 9. I was not doing research back then. But, yeah, and how do we do the binarization? Well, what they did back in the day, they just looked at the sign. So if it's a negative sign of the weight, it becomes 0. Otherwise, it's a 1. One way how you can do it. Good. This is how we do it. So the key thing is we're learning discrete variables through continuous optimization. Why do I say that? Because if this is binary and the activation functions are binary, that means that here in this representation layer, these neurons are either on or off. They're also binary. The inputs, also binary, and the outputs, also binary. Everything is binary, which is exactly what we wanted. So in embedded computation land, people were using it just out of necessity for making things efficient. They didn't want binary networks per se. They just, it just worked. But for us, this is exactly what we want. So we're going to take a leaf out of this playbook. Now, suppose we would do this plainly directly. I have to ask my favorite question. What could possibly go wrong if we would do it exactly the way that they did? Would we get really this pattern interpretability that we strictly want? It's better than nothing that we get, right? I mean, it's not going to be horrible, but is it going to be great or as good as we can get it? Can also give that one away? No. Does anybody have an idea? I mean, one of the points is hidden on the slides in a way. One weight matrix, another weight matrix. What does that mean? We have two weight matrices. That's what it means, obviously. But what would it mean in practice? They can be different. Suppose they are different. What does that mean? That means that seeing ABC might make me spit out DEF. Right? This is what we strictly allow here. Which may be something we want to do if we want to do rules or whatnot, right? Sure, but right now we don't want that. We want to have a strict interpretation that ABC, we haven't done, so if we see ABC here, then this one is on and that generates ABC. So it would be a bad idea to use two different weight matrices. We should use the same weight matrix, transposed, obviously, right? Because it needs to map from the same hidden dimension size to the same output, respectively, input space size. Binarizing like this, is this smart for us, for our setup? So here we were saying, okay, if it's below 0, up, off, otherwise it's on. Very harsh. Our data is going to be very noisy. So if we have a deterministic way of doing this, we might lose out on the opportunity to learn noisy patterns. We're going to be focusing on those parts that are most prevalent, but not those parts that should belong to the pattern, but not quite. We have a very strong inductive bias against noisy patterns. So we should not use something that is this strict. We should use something else. What that is, I'll show you in a few slides. So, different view on the problem. For us, this is now already natural. If I give you this record as input, beer and chicken, then I would like this neuron to be on, or another one, and that one reconstructs beer and chicken rather than something else. So then we can call this the dinner pattern, and so on and so forth. So, and then in the set that we have, in the end, we want to reconstruct the inputs. Now, this game we already played a little bit on the previous slide, but I thought it was more fun. So these binarized neural networks only learn -1, type of pattern. There's no clear, strict connection to conjunctive patterns. This is also where the noise fucks things up. Sorry. Nonlinearity, that is the -1. Nonlinear is great for expressivity, but not for interpretability. We want to have things strictly interpretable. So we want to get rid of that. We also do not want the network to memorize individual inputs, which these BNNs, they can definitely do. If you give them a wide enough bottleneck layer, bottleneck between quotes, then the first neuron can be learned, can be trained to be, some representation of the first record. Right? So this one has then, let's say, these 5 inputs, right? And reconstruct exactly, that is coincidentally exactly the first transaction. The second neuron does this, the second, and so on and so forth. We don't want that. We want these things to be really, truly patterns. There's no guarantee that the BNN will overfit like this, but we want it to slap around and avoid it from doing that. And standard reconstruction laws does not work, or not work well, on sparse binary data. This is already what we said about the signal that you get, right? That continuous value data data is like, if you have, let's say, x5 has 0.1 and your prediction is 0.2, okay, that is not great, but at least it's not 20, right? I mean, whereas binary, it's on or off. Yeah. So this is our proposal. I have to admit that I needed a long time to warm up to the name. I hated it for a long time. Right now I love it. Binaps, stands for binary pattern networks. This is already where it goes wrong in my head, because I don't think that names of methods should stand for something. This drive to have acronyms or backronyms, nah, right? I mean, like explain is kind of an acronym, but not really, right? And grab is not, and slim is not, and crimp, binapse is. But then I realized, hey, synapse, but now binary is binapse. And the moment that realization clicked, ever since I love it. But formally it stands for binary pattern networks, but you can forget that. Good. So what are we going to be doing? We're going to be at first getting rid of having two of these matrices. Actually, no, what we're first going to do is binarize the weights. I mean, okay, there's going to be now between 0 and 1. So not -1 and 1. So there's really strictly on and off. So we're in a way even more harsh than the BNNs, but that's fine. So that means in the backward phase, the continuous weights are going to be restricted at 0 and 1. We really just clip them. They can't go above, they can't go below. And the inputs, and then, well, within this range, the moment we need to binarize them, we're not going to do this deterministically, right? -1 plus, or I mean above or below 0, right? I mean that, but what we're going to be doing, we're going to be interpreting the 0, 1 as a Bernoulli variable. So the closer a certain weight is to 1, and it gets some input, right? The higher the probability that it's let through. If this weight is very low, let's say 0.0001, there is a very small probability that this is let through, but otherwise no. And that serves a number of purposes. I mean, we go a little bit closer to continuous valued land, right? I mean, but just the probabilistic one. We get noise robustness, and we can pick up, we can sort of tune the weight down, 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 up, up, up. We can sort of figure out, hey, wait a second, A is not there all the time, but often enough, right? Or actually, it's really important A is there. So we have to push that really to 1, and so on and so forth. The other thing, I already gave that one away, the weight matrix, we don't want two weight matrices, because then you get this weird interpretation problem. No, just mirror them or transpose them, whatever you see or like better, right? I mean, that really means if we see something on the left-hand side with the same probabilities, we now project it out over the. Good. Then, yeah, and the activations, they also just become binary. Rather than -1, 1, this is just, everything becomes truly binary. Good. Now, we have one problem left. And there I have to say, this is then the proposed solution, but whether the solution in practice works really as intended, I'm not so convinced at this stage. I mean, let's say there's evidence both in favor and to the contrary. The idea being, suppose that we have a pattern ABCD, right? Length 4. And now we would like the network to be able to learn, well, at least 1, respectively 2, respectively 3, or all 4 need to be present before I let it go through. How do we do this in this setup? Because everything is now, well, in the forward phase is binarized. For this, we use a bias term. So every individual, let's say, neuron in the hidden layer gets a bias input. So some neuron that's always on and then another weight. And how we now interpret this is basically it subtracts that from the input that the neuron receives. So if the bias is very high, that means that, well, we subtract a lot, that a lot of the input needs to be on. If the bias is low, well, that means that at least one of these neurons is fine. We set a minimum value for these, for the bias. Basically, if you have a pattern that consists of at least 2 elements, you don't want to say, okay, I want to see 1 of those. So bias is basically set at least 2. That's the general idea. Whether this is, again, a great idea or not, I leave that open. Okay, so putting this together, the ideas that we now saw from the binarized neural networks and the ideas that we then introduced is that in the forward phase, so the reconstruction phase, we're given some binary input. We have our weight metrics, which we get Bernoulli values, right? I mean, that's so we flip coins all the time, right? That then we have the activation for or not, right? I mean, and then, well, again, we reconstruct again after Bernoulliing, and we get a reconstruction. Okay, that's when we can now compute the loss, right? Loss is computed in binary land, not in continuous land. But now, in order to tune the weights, we need to put on our continuous optimization hat, And all of a sudden, we pretend that we were not working with binarized weights and everything. No, they were just continuous. Effectively, we want to push up and down these probabilities. So it's a very probabilistic, almost Bayesian, not strictly Bayesian, almost Bayesian interpretation of a neural network. And then we can backpropagate. So you say, okay, wait a second, you turned this one on and it shouldn't be on, or you turned this one off and it should have been on. Then we just get our errors. We can backpropagate. That means we can update these weights. And because it's symmetric, it automatically means that we also flip it the other way around, right? So it's a very short backpropagation in a way. Yeah, then we clamp after the updates the weight metrics and the biases to make sure that they really are between 0 and 1, right? Maybe the backpropagation pushed it below 0 or above 1. We don't want that. And then we'll rinse and repeat. We do this in mini batches over all the data, and then we arrive at the neural network. Okay, great. So yeah, typically we use PyTorch with Atom, but your mileage may vary in that. You can also do like full batch or whatever you want. But for us, this works really well. Does this fly? We're going to be comparing to a few different methods. We're going to be comparing to ASO. ASO is a method for Boolean matrix factorization. So, like I said, matrix factorization for Boolean matrices, minimizing the Frobenius norm, basically between the true data, so the input data, and how it explains it using the factor matrices. So every error, every 1 in the data it does not cover is an error. Every 0 in the data that it turns into a 1 is also an error. So just symmetric. Slim, you know, don't need to explain. Desk, you don't know because I didn't get to explain it last week. Think of it as a version of Slim that is perhaps a bit smarter. Hyperparameters, well, Slim and Desk don't have any, so just run out of the box. ASO has a method called MDL for BMF that Pauli Miettinen and me developed that can automatically search for the rank and tune these things. So we used MDL for BMF, yay for MDL once again. Then for binaps, we have a number of hyperparameters, namely how large should the hidden layer be, and the learning rate. So well, we optimize this, and this is basically the values that we use. Again, are these optimal? Nobody knows, but they work well in practice. Let's first see, does it scale? Because that was our key goal here, right? Slim and friends are already very good if you have small data, but what if you have many, many features? So here we have a number of features going from hundreds, thousands, 10,000s, and or hundreds of thousands, sorry. No, this is tens of thousands of features. And for the smaller number of features, we use 10K samples. And for the larger number of features, we use more samples because, well, you need more evidence to be able to find patterns. And we use 10K here also to help our competing methods and make them a little bit faster because otherwise it's... What do we see? These are the F1 scores comparing the discovered patterns to the ones that we planted in this data, right? So this data is essentially random. We plant a number of patterns. We shoot a few holes in it to make it too easy. That's roughly how you can think of it. And what you see is that we have desk, well, theoretically very nice, but F1 scores, I mean, it starts to get better and better, but I mean, here it sort of breaks down because the runtime starts to become insane. So maybe it would have done this, but probably not. Slim actually does very well up till, well, the moment we go to many samples and also there with many more patterns. So ASO is really the stuff for nightmares for me. Because, so Pauli Miettinen, the author of ASO, and me, we did our PhD in parallel, and I was on the crimp and slim and compression, and he was BMF track. And well, let's say both of us have opinions. But we're also friends, right? So that's also an opinion. We are friends. But in his view, BMF is superior. In my view, MDL is superior. And we both had good arguments. And for a long time, I would say I had the better arguments. This is also why we have this MDL for BMF. And with this MDL for BMF, I kind of created my nemesis because now ASO is all of a sudden very good and better than Slim. So I kind of hate ASO too. What we see is that ASO actually does really well, gets very high F1 scores, is really good at recovering the true patterns, but it starts to completely break down after, let's say, a few thousand features. Basically, the runtime goes through the roof and it can't do anything. This blue line is binaps. And of course, when it's sadly, it's not at 1, right, in F1, but few samples, it's not good. Okay, why would you train a neural network with many, many parameters if you have little data, right? So I'm not too sad here. If we have a reasonable number of features and samples, okay, good, then it starts to be very competitive. And the moment that you have really an insane number of features and difficult structure, okay, then it starts to taper off, but it runs. This is how much time it needs. So let's start with ASO, right? You see that it just shoots up, and then, well, now we're at 10 to the power 5. So this is 10,000. This is, well, I think a day, pretty much. What we said is a maximum runtime. Slim, similar happens, but it's slightly faster than ASO. This is why I think it's superior, of course. Desk is again a bit faster, but not really better. And Binaps, as you can see, scales very nicely. And not quite linearly, right? So it's the one scale, the X scale are both logarithmic. So the fact that it goes up is a bit disconcerting, but it scales. That is good. I can show you all sorts of results with F1 scores on, or whatever, right? Or again, on text data. But we set out, can we now finally use pattern mining on this biological data? I said Jonas, bioinformatician. So this was really the goal of his PhD. And the answer is yes. So we use data from the, well, slightly misnamed 1,000 Genomes project, where we have genomes from 2,500 individuals. So either these bioinformaticians can't count or something else happened. I think they just simply got more money and could sample more individuals. Then the data is very rich. We chose to look at the SNPs, so the SNPs, right? These point mutations. There's also other things that we could look at, but just because it makes it more difficult to interpret. 228,000 features. So this is basically where ASO and all the others, they break down completely. They can't even look at this. Yeah. Also, you could say how we can downsample. Like, yeah, which sample? Would you use, let's say, the first 10,000 and then the second 10,000 and then say, okay, well, there's not going to be any patterns in between, or we have no idea, right? So there's really a need for something that can look at it as a whole. What we found are a number of really nice things. So you're not bioinformaticians, or at least not many of you, I guess. What we see is that certain patterns have, well, yeah, blocks of consecutive variation. This means that we have on the chromatin, we have, let's say, a mutation here and here and here, and so very close to one another. This is exactly how, well, biology works, because things get spliced in and out, right? I mean, it's very seldom just one individual mutation that drives. No, typically you need a set of things to really go from, let's say, dark hair to blondish hair, something like this, right? I mean, this is not scattered all over the genome. This is relatively compact, which makes sense, right, from biological processes. So that's what we find. We find alternate phasing. So if you have this mutation, you don't have that one, or vice versa, because having both at the same time is not going to give you a good time. We find patterns like that. We find patterns that link, well, developmental genes, and this is really cool. So we find, let's say, A, B, C, D, and they have known roles in like developing a cell, but together with genes E and F, which are not documented in the literature. But if you look indeed at the data, they're very strongly correlated to exactly this set of A, B, C, D. Individually, just to A, just to B, just to C, not so much, right? So that they basically were hidden in the noise is totally understandable. We gave these to our friends at HIPS and other bioinformaticians, and well, they're digging into them, doing lab tests and whatnot. We say, okay, wait a second, this was 2021, why isn't it done yet? There I have a small anecdote, like really a mini one. Alex, another former PhD student of mine, his, let's say, research internship in my group before he started his PhD. We worked on some bioinformatics project. We had a very cool result. They were very happy with it. Like, oh yeah, we're going to test this. Fast forward 4 years later, Alex is done with his PhD. Like, we have the results from the lab. Let's write a paper. This is a bit how slow these types of verifications can be, because there's actual people that need to request actual money to do actual work in an actual lab, and especially in this particular case, I mean, well, doing knockout experiments on humans is of course a bit questionable, right? We also have similar results on tuberculosis bacteria, which also something that, yeah, we can, we don't have to have so much ethical concerns about bacteria as for humans. But at the same time, if we now create a killer strain of tuberculosis, you can't really say to a Hiwi, okay, well, just try it out, right? Just sick it out. No, right? So it's a very careful procedure to have to walk through. But these types of patterns, that include known components and then few unknown ones are the ones that make me really happy, and it's one of the reasons why I'm doing what I'm doing. It's not specifically for bioinformatics, but there we have evidence like, hey, this makes sense and this we didn't know, let's look into this. The other way around, we have no idea what this is and this we do know. Yeah, there I would also say, okay, let's not touch it yet, right? I mean, until we understand a little bit better, because maybe we just found scrap. Like I said, we found a number of these types of patterns. This is pretty neat. From an off-the-shelf or almost off-the-shelf solution, where we took this solution for embedded neural networks, held it upside down and shook it vigorously, and all of a sudden this works. Is Binaps now God's gift to pattern mining? No, but it is one of the best tools we currently have. It tends to be a little bit too lenient in adding, let's say, A, B, and C's to a pattern that perhaps should be separate patterns. So right now we're trying to find the right stick to make it stop doing that. We have, at the same time, in other projects, we've made it find rules, what Louis has been doing. That is very nice. We're writing this up right now. We can now use this. Actually, no, we have another variant of Binaps where if now let's say these genes are split into two groups, a bit like what we did last week as well, right? The papers with mining and the papers with not mining. Let's say these are patients of variant A, patients of variant B, right? We know just by their phenotype, by the way that the disease expresses itself. We can now use a variant of Binaps called diffnaps to find the differences between these two groups. That has already been used in a number of groups worldwide, and well, people are very happy with it. But it inherits the same problems that Binaps has, so my heart starts to beat faster if we can find better ways to improve Binaps, because then we have lots of downstream tasks we can automatically improve. Now, diffnaps, in turn, we can use on top of, in a way, explain. We can now find these are that set of neurons that are active when these are active, and well, but not there. So that means that we can now really scale up explain to, well, explain naps. That's sort of like how all these things now connect, right? So whether you now care about supermarket basket data, if you care about bioinformatics and genes and maybe drug development and whatnot, if you care about malware analysis or neural networks or anything for my part, as long as you can transform your data into a binary matrix, think of pattern mining. Think of the idea of, rather than just optimizing accuracy, think of like, how can we maximize extracting knowledge? Taking this descriptive viewpoint. And that is, I'm not going to say a superpower, but perhaps that is the right word. It really gives you a unique perspective that if you don't think in these combinations, right, in this description, in these patterns, you will never come up with these ideas. And that is what we're seeing right now, that, well, we can write many papers where the machine was like, oh, wow, we didn't know this was possible. Yeah, this was just right in front of you. But yeah, that's always the case with ideas. Good. Yeah. Mining at scale, blah, blah. This we already discussed. That's it for today. I hope I delivered what I promised. I hope you enjoyed it. I did. Thank you very much. See you next week. Bye-bye.